Hello, assalamu alaikum, and welcome to everyone. In Imran speaking. So today we are going to discuss about analysis of raw materials in QC lab. Grossly, we can separate raw material analysis into four categories. Number one is identification, number two is assay or potency, number three is impurities, and number four is physical quality attributes. We will dive a little bit deeper right now into these kinds of analysis. So first of all, let's come for identification. So this is quite self-explanatory because identification means what? To identify that raw material. So maybe I am using paracetamol. I have purchased paracetamol, so I must test and confirm that this is actually paracetamol, not any other API or other kind of raw material. So for that purpose, we preliminarily use four type of techniques for identification. So first of all is chemical identification. So what is chemical identification? Chemical identification is kind of identifications where chemical reactions are used for identification purpose. For example, maybe a raw material sediments after a chemical reaction, or it becomes turbid, or it changes color in certain ways. These are all chemical identification. And number two, and most importantly, we use spectroscopy, different type of spectroscopic method for identification purpose. Here we preliminarily use UV, IR, AAS, GC, LC, TLC, XRD, these sorts of instruments for identification purpose. This is the most widely used technique also. Number three is chiral identification. That is being done using polarimetry, that means polarimeter. So chiral carbons or chiral compounds cannot be differentiated using any sort of spectroscopic method. Only using chiral spectroscopy we can identify this stuff. And the fourth one is actually physical quality. Physical quality can be used for identification purpose, like melting point, refractive index, viscosity, calorimetry. We all know that if a pure substance have a definite melting point, refractive index, so the substance we are testing, if that have that definite melting point or refractive index, we can say that, well, yes, this is this exact compound. Okay, so these are all for identifications. Number two is assay or potency. So firstly, we identified that specific raw material, well, it is okay. Now this is the raw material. But the raw material we are using, does it contains 100% or the complete amount of raw material? Or it has any other admixture, like 90% of paracetamol and 10% of sucrose or glucose or anything else. Okay, so we must confirm that it has 100% or more or less likely that complete amount of raw material. Okay, so to do this we have to perform assay. We use predominantly three techniques for assay or potency determination. Number one is titrimetry. So titrimetry, we all know that it can be manual or it could be automatic. Manual titration, in here we use burette, pipette and some sort of indicator to perform titration. And that manual titration can be replaced by potentiometry, where the same burette pipette is used using a manual automatic system, and generally no indicator is required here, and the result prints out automatically using that potentiometry or potentiometer. The second most important method is chromatography. We use generally LC, GC with different sort of detectors are required to perform chromatography and to perform assay or determine potency. Okay. Number third method is spectroscopy, like UV or IR. We could use UV or IR to also determine the potency of any sort of raw materials. Number three, here written is impurities, but first I would talk about physical quality attributes. That's number 4 on this list actually. So in case of physical quality attributes, we have description, particle size, water content, viscosity. The first description, what is like we have raw material, we must address its description, like is it crystalline or amorphous, what's its color, is it lumped or so. These are all part of any description. Number 2 is particle size. What is the particle size of this raw material? We can use different type of sieves to determine raw materials, or more accurately we could use a particle size analyzer to determine this particle size. Water content, this one is very important. The moisture content or water content, we could use Carl-Fisher titrator or oven and a balance to determine the water content of that raw material. This is very important also to determine that potency over here, because in most of the raw materials, except that exact raw material, we have content, a little bit content or a much more content of water or moisture. We must determine the water content to determine the actual potency of that raw material. Viscosity is another physical attribute that is also being reported for certain raw materials, of course. Number 3 that is written here, impurities. Okay, now impurity is one of the biggest concern of raw materials, because, well, we have determined its identification, potency, physical quality also, but the question arises that if this raw material has any sort of impurities or any sort of admixtures, that is not good for health. Okay, to address this, we have to perform several tests. Impurity comes from two major categories, like one is organic impurity and another one is inorganic impurities. Okay, talk about inorganic impurities first. Mostly inorganic impurities are heavy metals. So where does this heavy metals comes from? Well, we all know that a raw material or an API is produced from a precursor through different chains of reactions. Okay, to perform these chains of reaction, we must use some sort of vessel. Mainly it is made of SS vessels, stainless steel vessels. So in these vessels, these reactions take place. Now when the reactions take place, what happens that a fragment of those metals sheds and comes into that raw material itself. That raw material contains those metals. Now these heavy metals can be very toxic for human body, and we must confirm that the heavy metal content of any raw material is below the acceptable limit. Generally, heavy metals are identified and quantified by chemical analysis. Mostly these are limit tests. Also, heavy metal could be identified using AAS, atomic absorption spectroscopy. Now come to the organic portion. This one is much complicated than this, but I'll simplify for you. Firstly, we talk about functional groups. So different type of functional groups may come from those reaction pathways also, like nitrate, sulfate, or these sorts of functional group can come in the raw material, which is not intended for human consumption or any sort of consumption. So we must confirm these functional groups are within the acceptable limit. Mostly chemical analysis are performed to confirm the functional groups, okay, the quantity of functional groups. Now come to VOC. VOC stands for volatile organic compound. So it suggests that this is an organic compound that is volatile. So where does this volatile organic compound comes from? Well, we all know, as I said earlier, that the raw material we are using is produced from a precursor raw material. Now from that precursor raw material, we use different type of chemical reaction pathways, and to perform those chemical reactions, we must use some sort of solvent. It may be water, it may be ethanol, methanol, tetrahydrofuran, acetonitrile, any sort of any number and any sort of liquid reagents. So these liquid reagents are not acceptable. Not acceptable, I means that liquid reagents, some of these are highly carcinogenic, and these are not acceptable for human consumption. So what happens that these liquids actually may come in the final product during these reaction chains. So we must test in QC lab to confirm that these volatile organic compounds are also within their acceptable limits. Generally, this VOC is determined using gas chromatography. Now come to impurity and related substances. These two terms may be used interchangeably, but first I might talk about related substances. So related substances are the substances that are actually related to that raw material we are using. So what happens from a precursor becomes a finished raw material through different sort of analytical pathways. Different type of chemical pathways. So what happens? A becomes B, B becomes C, C becomes D, D becomes F. That is the final product. Now all this A, B, C, D, these are unwanted for our human consumption. So these ABCD may come in the final product F, and that is unwanted. So we must confirm that these ABCD or whatever the precursors are, these related substances are within their acceptable limits or as low as it is possible. So these are called related substances. This is one sort of organic impurity. Now, organic impurity could be directly said that related substances also accept those related substances. Other form of organic impurity, if we get from that raw material, these are called impurity. These are generally unknown impurities. We don't know the origin of those impurities. These are not the precursors, but it can be produced during transportation, during any sort of treatment, or maybe it comes from environment or so. We must confirm that this impurity and related substance both are within their acceptable limits. To test impurity and related substance, mostly HPLC or liquid chromatographic systems are used. So these are the four major categories of raw material testing with their subcategories. Also, we have microbiology. In microbiology, we test a pathogen, that means that a viable bacteria count, how much these are, and also how much toxins are there in the raw material. It may come from environment or product, some sort of products. So that's all for today. This is the raw material testing and quality attributes. Hope this clears a lot, and in the further videos we will discuss in depth of these analytical pathways and how these are done in any sort of QC lab. Thank you.